Welcome back. Today we continue our series on memory. Memories are made of this, and I entitled this talk, A Breakthrough in Alzheimer's Disease. Just to set the stage, Alzheimer's disease is a tremendous problem. It's an exponentially increasing problem that has worldwide implication. In the first slide here I have printed a text from the World Health Organization and two URLs that you might want to visit. The message thus far is that currently about 48 million people are living with dementia, mostly of the Alzheimer's type, and 7.7 million new cases are diagnosed each year. If you boil this down, you can determine that about every 4 seconds a new case of Alzheimer's disease is identified. Now, not all dementias are the same, although they share very important biochemical and cell biological features. As you can see, Alzheimer's disease is listed here, and it's by far the most common dementia in man. There's a frontotemporal dementia which has different manifestations, usually bizarre and unusual social behavior. There is vascular dementia, which has a stepwise progression, also called multi-infarct dementia, progressive supranuclear palsy, and finally Lewy body dementia, which is characterized by prominent visual hallucinations. If you see visual hallucination in an elderly or even middle-aged person, Lewy body dementia is a high index of suspicion. Now, we have made progress in medicine in recent decades. You can see here that the mortality for a number of diseases has decreased. Prostate cancer, for example, heart disease has experienced a dramatic, sustained, and significant decrease. Stroke, likewise. HIV, with the advent of cocktails of antiviral drugs, basically has been converted from a lethal disease into a manageable chronic disease. The exception is Alzheimer's disease with a 72% increase in mortality. So those are the odds. So let's dig a little deeper and see what actually is Alzheimer's disease. It's characterized by the deposit in the brain of some unusual protein aggregates, such as the amyloid plaques or neuritic plaques and the neurofibrillary tangles composed of so-called tau proteins. The talk today will be mostly about the structure right here, the amyloid deposit. Amyloid deposits have been known for an extended period of time and have been characterized in great depth. It was thought that perhaps amyloid is the key perpetrator that sets in motion a progression of changes in the brain leading to dementia and finally death. There has been some doubt recently whether amyloid is really the key player. Amyloid may in fact be an innocent bystander, and the real culprit may in fact be tau. Amyloid may trigger an initial disruption in the brain, leading to the accumulation of phosphorylated tau, which is the real toxic and lethal agent. We will concentrate on amyloid, and in the next slide, I show you the so-called amyloid hypothesis. So here it goes. Amyloid is a membrane protein that can be released from the membrane by a number of enzymes, which we will investigate shortly. So the so-called A-beta monomers tend to aggregate in the brain, and these aggregates form fibrils, become insoluble, and are deposited around the nerve cell. In response to this injury, tau aggregates are formed, which eventually lead to neuronal death and the death of the patient. And the next slide. shows you really the key smoking gun, the evidence that amyloid is a key player. So the amyloid protein has been characterized, the genes have been cloned, and multiple families have been identified who have molecular genetic polymorphisms in their sequence coding for the amyloid protein. And all the red little circles that you see here are known mutations associated with an early onset of Alzheimer's disease transmitted genetically in families. Now, this is very powerful evidence. On all these cases, you get abnormal processing of this huge protein that's in the membrane of nerve cells that releases a certain piece of the protein called APP142. It's a 42 amino acid long peptide that is cleaved from the original intact protein by the action of a number of enzymes that cut this membrane protein into pieces. And some of the mutations that lead to Alzheimer's disease are in fact found in these enzymes. For example, here, here, and here. So these are enzymes co-located in the membrane whose job it is to turn over the amyloid protein, cut it into pieces so that new proteins can be inserted at the appropriate time. So both mutations in the amyloid gene as well as in genes coding for the peptidases that cut the protein into pieces are involved in familial Alzheimer's disease, strengthening the case that amyloid is a key player in the initiation, at least, of Alzheimer's disease. The next slide is a more simplified cartoon. You see here that the C-terminus is in the membrane. It's cleaved by a protease. The beta C-terminal is remaining in the membrane. And the amyloid beta is released. Amyloid beta tends to aggregate, and it wasn't really clear until very recently what exactly mediates this aggregation, this tendency to form insoluble clumps of protein aggregates. And only in the last few months has the physical nature of this interaction been exactly described. Next slide. So because of its role in the etiology of Alzheimer's disease, we want to know the exact molecular structure at the atom-to-atom level of the amyloid beta 142. And this finding has been eagerly awaited and is now available and was just published in the Proceedings of the National Academy of Sciences. And I give you a quick overview over what has been found. So here then is the near atomic resolution of a model for the AP142. The amino acids in red here indicate sites of mutations that I showed you previously that are involved in the familial transmission of Alzheimer's disease. Now the structure of the individual beta amyloid chain is such that it tends to have hydrophobic surfaces that tend to clump together, exclude water, and become impenetrable. And here you see what happens. You see a lattice of beta amyloid clumping on top of each other and become nearly insoluble, even with strong detergent action. So that seems to be the mechanism. We now know the exact molecular structure of the event. In the next slide, you can see a more dramatic presentation of this. You see the so-called beta sheets that line up in a plane. These beta sheets clump together through hydrophobic, water-excluding interactions, which are extremely difficult to break. Very recently published in Proceedings National Academy of Sciences in 2016. And in the next slide, you see a finding described here in Nature Physics, one of the Nature journals. Physicists became interested in this mechanism and have generated computer models that predict how this event happens. So you start out with an oligomer, which eventually forms a fibril. And these fibrils act like a seed. You can attach small oligomers, monomers and oligomers to this fibril, which becomes a catalyzing agent to lead to the production of new seeds for the nucleation of additional fibrils, which then are a progressive avalanche of amyloid deposition. So that is the trick. That's how amyloid, beta amyloid 142, becomes so destructive to the brain. That sounds like a very convincing theory implicating amyloid beta 142. There's a problem, however, and it is that the clinical picture, the degree of dementia and cognitive impairment, does not correlate well with the degree of amyloid deposition in the brain. Rather, it correlates much more strongly with the deposition of phosphorylated tau, which is a downstream event from the amyloid formation. So these findings are positive, but they certainly do not tell the whole story. We are missing essential intermediary steps in the sequence of events. The next slide. Now, when the amyloid is deposited, all hell breaks loose in the immune system. The brain microglia, which surveys the state of health of the neurons, will be alarmed by these oligomers. There are in fact receptors on microglia cells which will interact with these oligomers, take them into microglia and activate the microglia. The microglia then becomes highly agitated and tends to accumulate around amyloid deposits, trying to remove them by something called phagocytosis, eating the amyloid plaque. And that seems to be a mechanism which we can tap into to remove the deposit of amyloid from the brain. In the next slide, you see some representation of activated microglia. In panel B, for example, you see the bright green fluorescent cells. This is activated microglia, in this case in a brain from a transgenic mouse, which carries a Alzheimer's producing gene in its genome. You can also see it in panel D, which is from the section of another transgenic mouse. And in panel A and C, you see actual sections from human postmortem brain of a patient that had died of Alzheimer's disease. The next slide. So here then is the sequence in summary one more time. So the amyloid cascade hypothesis says that because of mutations, missense mutations in the precursor protein or in the presenilin 1 or PS2 genes, which are the proteases processing the amyloid, you get increased A-beta 42 production. This then leads to oligomerization and the precipitation of insoluble amyloid in the brain, as I showed you the articles in PNAS and Nature Physics. This then leads to the activation of microglia and to the increasing injury to synaptic connections, eventually to the alteration of kinases which hyperphosphorylate a protein called tau, which is a microtubulin-associated protein, the cytoskeleton of the neuron, keeping the neuron in a certain structural stability. And then this finally leads to the death of the neuron. So this is then the amyloid theory. Now, therapeutically, if we could go in early in Alzheimer's disease and remove amyloid, perhaps with an antibody, and what I have in mind is a so-called monoclonal antibody. These are antibodies that have been trained, so to speak, to recognize only very specific sites or epitopes of a protein and do not cross-react with other proteins in the brain. If we could get such an antibody into the brain, perhaps it would tag or bind to the amyloid and then solicit a response from microglia, leading to the disruption and elimination of amyloid plaques. That is the theory, that is the therapeutic strategy. And it's not for lack of trying. Next slide will show you some other complications though, because amyloid, of course, does not live in isolation. And we know that a number of risk factors can lead to the increased risk for amyloid deposition in Alzheimer's disease. For example, apolipoprotein E, which is a common allele in human populations, will increase the risk of Alzheimer's disease several-fold if you have both genes for apolipoprotein type E. And many other factors have been identified, such as lymphatic flow in the brain. It's a very recent finding. Mitochondrial damage, reactive oxygen species, and disrupted sleep, which is a common phenomenon in the elderly. Disrupted sleep tends to lead to a disruption in neuronal activity in such a way that increased beta amyloid is produced, which in turn disrupts sleep even further, leading to a positive feed-forward loop, which eventually can aggravate or perhaps even precipitate Alzheimer's disease. So there is a host of environmental factors, and perhaps it is those that are most amenable to intervention at this time. Another very powerful factor is obesity or the metabolic syndrome. As I've told you in the metabolic syndrome lectures, we are facing an avalanche of metabolic syndrome in the world, with about 1/3 of Americans now qualifying for the diagnosis of obesity. Obesity, of course, is a precursor to diabetes, hypertension, and other problems like dyslipidemia, all of which are risk factors for Alzheimer's disease as well. So now let's go to the proposed intervention. We will concentrate on one aspect. You can see here beta secretase inhibitors, other inhibitors of the proteolytic processing of amyloid have been developed, but we want to focus on trying to eliminate the oligomer or monomer for A beta 142 with a specific antibody as indicated in the red box. So the problem, of course, is number one, create a very specific antibody to beta amyloid 142, get it into the brain, and have it work in such a way that it triggers the phagocytotic removal of amyloid by activated microglia. And it's been tried a number of times with stunning lack of success. So here's a trial that was published in the New England Journal of Medicine in 2014, a negative trial. Here was a monoclonal antibody that failed to make any significant clinical impact in Alzheimer's patients. And here is another trial of another monoclonal antibody, likewise failing to make a significant impact in Alzheimer's disease. In comes yet another monoclonal antibody, and this paper was published just a couple of weeks ago in Nature. Now, Nature doesn't usually publish clinical trials, but it published this because this particular antibody, aducanumab, reduces the amyloid plaques in Alzheimer's disease with a vengeance. And it doesn't really cause any significant side effects. Some of the other trials were troubled by side effects in the brain, such as inflammation, that either led to termination of the trial or limited the amount of antibody that could be used. So here you see the key picture in this paper. On the left-hand side, you see the baseline imaging of the amyloid deposits in reddish or yellow colors. An imaging agent has been found a number of years back, so we can now probe the brain with PET scanning and have a ligand label the amyloid deposits in the brain directly. And you can see here that on the right-hand side of the panel, the placebo and then an increasing dose range from 3 to 10 milligrams drastically eliminated and reduced beta amyloid deposition in the brain of these patients. So there was a striking dose-dependent reduction in the PET measurements of A-beta plaques in the brain. This has never been seen before to such an extent. Now, in preclinical trials, the researchers have shown that this antibody, in fact, penetrates into the brain. Only a small fraction, however, of the administered dose actually winds up in the brain because of the effect of the so-called blood-brain barrier. But for some magic, substantial portions of the antibody tends to get in and apparently elicits the response that is required to solubilize and remove amyloid deposits from the brain. Now, here is a numerical description of the effect, and you can see that the antibody leads to a significant reduction in the signal that comes from the PET scan in week 26 and week 54 for different dose ranges of this antibody. Again, an effect that has never been seen in this powerful of a fashion. Now, this trial was not powered statistically to look at the clinical impact of this. Does the removal of the amyloid plaque in fact lead to a decrease in progression of the disease? Does it slow the disease or even improve cognition in these patients? Nevertheless, here you see some findings on a rating scale of dementia. And you can see here on the right-hand side, with placebo, you have a significant increase in the rating scale, and then in a dose-dependent fashion, you see a decrease in dementia rating according to how much of this antibody was given to the patient. So that's very powerful evidence. Not only is the antibody better than placebo, but it does so in a dose-dependent fashion, which you would expect for a biological effect. Another way to look at this is to look at the very commonly used mini mental status examination. A brief bedside 20 or so item examination asking for orientation, following simple commands, etc. And you can see that placebo led to a further decrease in cognitive skills. The mini mental status score continues to go down, whereas in different doses of the antibody, for example 10 milligrams, the decrease in the mini mental status score was much less pronounced, suggesting that the progression of the disease had been slowed quite significantly. Now again, this is not powered statistically to prove this once and for all, and follow-up trials certainly are needed to come to a conclusive finding whether or not this antibody can slow the disease and have any meaningful long-term impact on the lives of patients. Now, how about the activation of microglia? Does this antibody live up to expectations? And you can see, yes, indeed. The microglia here is increased. Now, this is in an animal model in mice who carry the Alzheimer's producing gene. And you can see that plenty of amyloid eating microglia is accumulating around the amyloid plaque. So we can break down the data into two groups. the removal of plaque less than 1 standard deviation from control or more than 1 standard deviation of control. Basically dividing the patient population and testing whether the impact on cognition is really dependent in this way on the removal of plaque. And you can see here that more than 1 standard deviation makes a much more significant impact on a decrease in the ratings for dementia and leads to a significant decrease in decline as assayed by the mini mental status examination. In controls, you see a dramatic decrease in mini mental status score, and in the high dose of the antibody, in those patients which had the highest removal of amyloid plaques, you can see that the mini mental status scores were maintained at the level prior to the intervention. So that's the positive news, and it's a real breakthrough. But it's not the only breakthrough that we need, because unaddressed at this point is the issue of how progression occurs from amyloid deposition to phosphorylation of tau, which probably is the more critical protein for the eventual destruction of neuronal circuits. But there's quite a bit to celebrate here. This has never happened before. Now, will this make a huge impact in clinical practice? Probably not. Monoclonal antibodies are extremely expensive, and it's difficult to fathom how to treat 48 million patients currently known to have Alzheimer's disease with a very expensive antibody. And it's even more difficult to imagine if you have a new Alzheimer's patient every 4 seconds, how you can make and sell and pay for enough antibody to make a real impact in the lives of these patients. However, as proof of principle that amyloid is a critical element in Alzheimer's disease, this is really a significant piece of progress. So thank you for your attention. We will revisit this again when we give a presentation more specifically targeted towards the physiology and the impact of tau in Alzheimer's disease.